Let's see the important points in the chapter Sexual Reproduction in Flowering Plants. The sexual reproducing part, that is flower, contains both male as well as female parts. When we say female, then we have stigma, style and ovary. When we say male, we have filament and anther. So now let's learn about the structure of female reproductive parts in a flower. Stigma is a landing platform for the pollen grains to fall, and even it helps in germination. Whereas when we talk about the style, that is used for development and growth of pollen tube, as well as male gamete might undergo mitosis, cell division. Whereas when we talk about the ovary, it is the large bulging portion at the base, and it is the one which contains ovule. Ovule is the one which is going to transform into seed in the future if it has got fertilized. Let's talk about the ovule in detail. The ovule that we study over here is called anatropous ovule, and it contains two layers or coats which are called as integuments, outer integument and inner integument. Inside that there is a ball-like structure which contains a cells called nucellus. Okay, out of that nucellus, one of the cells transforms to become a megaspore mother cell. Megaspore mother cell undergoes division, that is meiotic cell division, to form 4 cells, and for this all the 4 cells are haploid and are called as megaspore tetrad. Out of the megaspore tetrad, 3 of the cells degenerate, whereas one cell again transforms to become a huge cell which is again called as embryo sac. So now the nucleus which is present in the embryo sac is an haploid nucleus. So it has only a number of chromosomes. This haploid nucleus undergoes 3 times mitotic division to form 8 nucleus. These 8 nucleus are present, and that's why the embryo sac can be called as cenocytic cell. Cenocytic means having more than one nucleus. Out of them, 3 of the nucleus move toward the chalazal end, 3 of them move toward the micropyle end to remain in the center. The 3 which move toward the chalazal end, they undergo cytokinesis to form antipodal cells. Similarly, the one that were toward the micropyle end undergoes cytokinesis to form egg apparatus, which contains one large egg and 2 synergids. Synergids contains finger-like projections called filiform apparatus, which helps later to show the pathway for the pollen tube as well as pollen, male gametes to fuse with the female gametes. So that's about the embryo sac. Now we can see that there are 7 cells and 8 nucleus inside the embryo, and this is what is mature or embryo, and it is ready for fertilization. So it is waiting for the male gamete to come and fuse. So that's what is the structure of the female reproductive system. Let's now learn the structure of male reproductive system. So male reproductive system in the plants consists of an anther and a filament. Filament is the stalk which holds the anther on the top, and there are 2 types. One is having just 1 lobe and another is having 2 lobes. So unilobed anther and bilobed anther. Mostly we study bilobed anther. Each lobe contains 2 chambers, and they are called microsporangium. So inside the microsporangium, pollen grains are formed. Let's learn the structure of microsporangium. So immature microsporangium consists of the outermost layer which are barrel shaped with a cuticle called as epidermis. Inner to that there are cells which are called as endothecium, and then another 2 layers of middle layer, and then inside middle layer there is a thick and dense cytoplasma containing with more than 1 nucleus called as tapetum, and this is the most important. Many a time this question has come in many of the entrance exam as well as internal exams. So we can understand what is tapetum. Tapetum are those cells which are present inside the middle layer cells. They provide nutrients to the cells because they have more than 1 nucleus. They show cenocytic condition as well as they have dense cytoplasma. Whereas inner to the tapetum there are cells called as sporogenous tissue, and these sporogenous tissue multiply by mitotic division to increase its number. But once the numbers are more enough, then their name can be called as microspore mother cells. Now these microspore mother cells undergo mitotic division. Now they undergo meiotic division. Now we cannot say mitotic division because unless it undergo mitotic division, we call them as sporogenous tissue. You will not find any difference in ploidy. Both has to end, but only the name we change because next step they are going to go for either mitotic or meiotic. Those cells which undergo mitotic division are called sporogenous tissue. Those cells which undergo meiotic division are called as microspore mother cell. And then these microspore mother cells will divide and form 4 cells. All the 4 cells are attached to each other, forming a microspore tetrad. Later each cell separates from the main and then become a pollen grain. So let's learn the structure of a pollen grain. Normally a pollen grain consists of 2 layers. The outermost layer is called as exine, whereas innermost layer is called as intine. Exine contains a toughest organic substance called sporopollenin, because of which it can prevent the pollen grains from unfavorable condition. So it might last for long, and those pollen grains who don't have this sporopollenin might be available for very long, very short period of time. Okay, so that's why we find some pollen grains survive for very long and some don't survive for very long, so that we can understand why it is so. Okay, that is due to sporopollenin. Though there is sporopollenin, but we find at a point there is no sporopollenin, and that point is called germination pore or germ pore. And from that germ pore, the pollen tube germinates when it is on the stigma. Okay, so now initially there is one nucleus in a normal pollen grain, but later when it starts maturing, it undergoes mitotic division to form two cells. So two cells, whereas one cell is very large with a large nucleus and another cell is small with a small nucleus. And the large cell is called vegetative nucleus, whereas the small one is called germ cell or generative cells or male gamete. So these male gamete take the nutrients from the vegetative cells. So this stage is the mature stage of a pollen grain. Whereas sometimes we find that the pollen grain even undergo that male gamete that is present inside may undergo one more mitotic division and form two male gametes. That means 3-cell stage might be the mature stage in some cases, or most of the cases 2-cell stage is the case. So if it is 3-cell stage, there is no mitotic division in the pollen tube. Whereas if there is 2-cell stage, then the male gamete must undergo a mitotic division in a pollen tube and become two more male gametes. That means in a dicot plant or let's say angiosperm plants, we find that there are two male gametes when they are reaching the ovule. Let us discuss about the pollen pistil interactions. After the pollen grains get mature, then they are dispersed from the anther because the pollen grain, I mean to say anther get dehiscence from the line of dehiscence. There is a line of dehiscence over there in the anther which will break and the pollen grain will spread. So here we have pollination. Pollination means the transfer of pollen grains from anther to the stigma of a flower. So there are two types of pollination. One is called as self-pollination, that we know if the pollen grains are from same flower or same plant, then it is called as self-pollination. But if it is from one flower to the another flower of different plant of same species, then it is called cross-pollination. If it is different species, they may not germinate, so that's not called, okay? So that's why here we call as cross-pollination as the transfer of pollen grains from one flower to another flower of different and plants of same species. So that's called cross-pollination. We can also have one more called as genetogamy. Genetogamy means it's also a kind of pollination where we say genetically they are self-pollination, but physically they are cross-pollination. That means we find two types of flower, male flower and female flower in a same plant. In that case, there is genetogamy, means transfer of the pollen grain from one flower to another flower of same plant. So genetically they are identical, but physically it is a cross-pollination. So this is about pollination, and for pollination different pollinators helps. So we know as per the pollinators, flowers get modified. We know that entomophily means pollination by insects. Terpetrophily means pollination by bats. Ornithophily means pollination by birds. If we talk about anthrophily, it's pollination by human beings. We know it's hydrophily, obviously pollination by water, and nemophily pollination by wind, and nemophily pollination by animals.